Okay, so what about this internal heparan sulfate proteoglycan environment? This is a gel-like substance, and it has properties that are neither perfectly aqueous nor perfectly solid. In this experiment, the large secretory granules from beige mouse mast cells were isolated and actually patched with a patch pipette, and a voltage was applied between the inside of the pipette and the inside of the vesicle. And under conditions where that voltage is positive, there's no change in the size of that secretory granule. But when the voltage becomes negative across the secretory granule, you can see that it begins to swell and extend, and it's so extended that it's very hard here to even see the borders. So if one monitors swelling, with a positive potential, there's very little change, and when a negative potential is across it, there's very high swelling, as if osmotic forces in this diode-like behavior might also be at work in helping to mechanically overcome some of the forces which maintain the stability of the lipid bilayer in the fusion process. All right, so how does a neuron regulate the contents of a vesicle? If there are transporters present, how do we know how much goes in? Because clearly the amount of transmitter inside a vesicle is very important. We've gone to great physiological extent, an effort to measure the size of a postsynaptic response and found them to be quite uniform. So how does this get set up? In this experiment, Mu-Ming Pu and colleagues took advantage of the fact that they can create in tissue culture cells which form very early synapses from neuron to muscle in 24-hour-old Xenopus embryos. And under these conditions, they can manipulate the genome being expressed either by neurons or muscles in this cultured system and record from the muscle as these very young motor neurons innervate them. When they transfect neurons with an empty vector or cDNA containing the vesicular acetylcholine transporter, actually this is just the cDNA without that, so this is the negative control. So both of these bottom traces are controls. They can record spontaneous activity in the embryonic muscle, and they can decrease calcium concentration to the point that they can look for the average size of a spontaneous miniature end plate current. So this is all voltage clamp, and without any expression they get an amplitude for their currents and a frequency. When they do, though, express the vesicular acetylcholine transporter, cDNA, in the neurons, there is a huge increase in amplitude and actually an increase in frequency. And we can see that in this column where the expression is only in the neurons, and in this column which is frequency with expression only in the neurons. Expression in muscle did not change the size of the miniature end plate currents. So this had no effect on receptor number, but quite a bit of effect on the amount of transmitter released, suggesting that the amount of neurotransmitter packaged in a synaptic vesicle is dependent on not the gradients that are being developed, but rather the number of transporter molecules functioning to move neurotransmitter into the vesicles. It also suggests a very interesting kind of plasticity that the system might utilize if it needed to change presynaptic release properties. So how is the vesicular acetylcholine transporter regulated? Turns out that there were two very interesting mutants in the mid-90s of C. elegans in screens that looked for motor abnormalities. There was one particular mutant called UNC17 and another one called CHA1, I think. And both of these had very similar uncoordinated phenotype. They would hatch, but they don't move or eat. They would end up starving to death. And separate labs investigated each different mutant and independently traced the mutant through the genome back to the same stretch of DNA. One lab found that the UNC17 coded for the vesicular acetylcholine transporter, or had a mutation in it, and the other lab found that the CHA1 had a mutation in the choline acetyltransferase gene. That's the synthetic enzyme that takes acetate and choline and merges them together to make acetylcholine. And they found, both of them, that both genes were expressed under the same upstream regulatory pieces of DNA, the same promoter operator region, or the same operon, if I have that correctly. Not only that, this was true in C. elegans, as well as in rat, and as well as in the human genome. There appears to be a natural single regulatory piece of DNA for both the synthesis of the neurotransmitter and the neurotransporter that's going to package that neurotransmitter into the vesicles, so that if there's a need to upregulate the vesicular contents, both are upregulated or downregulated in parallel.